I'm Ana Moles, and I have the pleasure to introduce Professor Dina Tiniakos. For those who don't know her, she is a professor of pathology and director of the Department of Pathology of Aretaion Hospital Medical School, National and Kapodistrian University of Athens, Greece, and a visiting professor at the Translational and Clinical Research Institute, Faculty of Medicine of Newcastle University. She is also the past president of the European Society of Pathology and current chair of the the ESP Advisory Board. So her main research interests are steatotic and autoimmune liver disease and hepatocarcinogenesis. And she is leading and co-leading several European and international research consortia on liver pathology, including the LITMUS. So it is with great pleasure that I introduce an excellent researcher and a good friend to you, and I give her the word so she can do her talk. Thank you very much.
Today we will talk about the origins from the origins to the future of metabolic dysfunction-associated steatotic liver disease. These are my disclosures. 6 months ago, I would have named the talk on how we got from non-alcoholic fatty liver disease to metabolic dysfunction-associated steatotic liver disease. I thought that we will start with this because there has been this change in the terminology agreed by 3 major societies and hundreds of people who did a consensus and a steering committee, and we're very happy that there were 2 pathologists in the group. Usually you don't see this in a type of consensus, so we're moving forward with this. And one from the ASLD, and I was the one from EASL. So I know how much work it took more than 2 years to come to this agreement and to refine everything. And we came up, and most people agreed with this new terminology, steatotic liver disease. Stear in ancient Greek means fat. So, and this is the word actually we use when we describe the disease in pathology. It's using this term. We never write fatty liver disease in our pathology reports. We write steatotic steatosis and steatohepatitis. So for the pathologists, it was not such a big change. So according to this new nomenclature, the previously called NAFLD is a metabolic dysfunction-associated steatotic liver disease, and the progressed form is the metabolic dysfunction-associated steatohepatitis. And alcohol-associated liver disease is a separate category. And we have also specific etiology of steatotic liver disease, when it is drug-induced or when it is inherited, and some other causes, and some cases remain cryptogenic. The most important part is that we recognize we always knew about these patients, but now they're officially recognized that they may have muzzled, as we will say, but may also have increased alcohol intake, but not so much to be called alcohol-induced liver disease. And these are the thresholds of alcohol that we're using. So starting with this, we can move on our main topic. The MASLD is a complex metabolic liver disease. It is considered a hepatic manifestation of the metabolic syndrome. It's mainly related to abdominal obesity and also diabetes, but the main features of the metabolic syndrome, if you have 3 of those, you are considered to have it according to the most accepted definition. So people may have hyperglycemia, dyslipidemia, and hypertension. 3 of those give you the badges of metabolic syndrome. So nowadays is the most common cause of chronic liver disease worldwide, not only in adults, but also in children. And this is the worldwide estimated prevalence, which is about 25%. But according to the map, you can see there are areas like the US or Australia and other areas, India, that may have more than 30% prevalence of MASLD. And here in Spain, we have the 20 to 29.9. So this prevalence parallels the increasing prevalence of obesity and type 2 diabetes mellitus, which they go together most of the times. what is the spectrum of muscle? What do we see under the microscope? So all the patients have steatosis. And when we say steatosis, we see in the hepatocytes these light globules of fat. And some people may progress to steatohepatitis. The numbers differ between the different countries and different areas in the world. You see it's more common in North America, less common in Asia to have steatohepatitis, but the mean is 1.5 to 6.5% globally of the patients who have MASLD. And 40% of people with MAS, as we will call the progressive form, may have progressive fibrosis. And these people may end up having cirrhosis. 3 to 5% may have cirrhosis at the end. But because it is such a big a number of patients who have MASLD, then this 3 to 5% becomes a large number for the health systems to deal with. And of course, in the background of cirrhosis, you may have development of hepatocellular carcinoma, which is like up to 11% of the cases of MAS with cirrhosis. But in MASLD, it's important to remember that we may have hepatocellular carcinoma develop in non-cirrhotic MASLD, like when you have earlier stages of fibrosis, as we have seen in many cases. So this is a disease that actually it has been called in the paper as an epidemic, and there were many papers for a call of action, and yet just to see the numbers, it is expected the prevalence to increase by 63% until 2030. This was a paper from the US, so you can see that more than 100 million individuals worldwide may have MAS. Fibrosis also may increase, double by 2030, and it is estimated that we may have up to 800,000 MAS liver-related deaths by 2030. And of course, we need recently to think about the costs, and there is a lot of cost to the health system having people with MAS, of course, in the next 2 decades, in billions of dollars. But is this a new disease? Well, MASLD has been overlooked for a long time, and the term MAS, or NAS, did not exist in the medical vocabulary until the 1980. But MASLD was familiar only to a small group of experts until 30 years ago. And if we see the number of publications, how they have increased from 1910 to 2019, you see the explosion of literature on MASLD, and also this is for fatty liver or steatotic liver, and this is for NAS or MAS. So there is a lot of interest for the disease now because it is so frequent. And the other important thing is that in 2023, like until last year, we didn't have any MAS-specific drugs or reliable biomarkers. This year, in 2024, we have a specific drug, and we may have 1 or 2 more during the year. This is the resmetirom, which was approved by the FDA only last month. And this is for treating patients with non-alcoholic steatohepatitis, MAS, as we say it now, reducing steatosis in hepatocytes. So, but still we do not have reliable biomarkers to diagnose steatohepatitis. We may have, as we will see, more reliable biomarkers to diagnose advanced fibrosis, but not to diagnose the progressive form of the disease. So what I'm going to do in the next 30 minutes, or maybe it may take a bit longer because I'm talking slowly. I will take you to a journey from a nebulous disease of unknown origin in the 19th century when it was first discovered, but we will see that we knew about that from antiquity, to the most investigated liver disease today. And pathologists like myself still hold the compass to navigate MASLD, because still liver biopsy is the most accurate method to diagnose MAS, the progressive form, and also to classify MASLD. But this may change, and we hope to change in the next near future. So what we know about Mazdelt before the years of autopsy, when the first pathologists could see what was happening under the microscope? Well, in the ancient times, obesity was a sign of success, prosperity, good health, and fertility in women. And we know that from the 30,000s BC, from ancient Greece, like prehistoric Greece, and also in Egypt. It was a royal and very important thing to have obesity. And obesity was first regarded as a pathological state by the Indian physician Susruta in India, that's 6th century BC. And Hippocrates from Kos, the father of medicine, recognized obesity and the importance of healthy nutrition. He writes a lot about the healthy nutrition for obese people. And Galen of Pergamon, now we are in AD, he thought that obesity was an illness, he called it polysarkia, and sarka means flesh in Greek. And he suggested diet and exercise, what we do today for most Mazdelt patients. The thousands of years passed, and we are in 1825, and Pierre Louis in France made the earliest use of the term foie gras, meaning fatty liver, in one of his books. And in 2036, Thomas Addison from England, the well-known Thomas Addison from Addison's disease, he was the first to use the term fatty liver. He actually was born in Newcastle, my other place. And he wrote that deposition of the adipose tissue in the liver parenchyma, omentum, and the mesenteries was the etiology of hepatic steatosis, also in children who have gourds with food. So they knew about this also in the children, because they were based for findings from autopsies. And they mainly did autopsies from adults who probably had also alcohol-related liver disease, but some of them could have had Mazdelt as we know it today. And children did not have alcohol, but he saw the same finding in children liver. In 1839, Karl Rokitansky from Austria, who was is like one of the forefathers of pathology, he wrote about the causal relationship between the hepatic fat and fibrosis. And in 1857, George Budd in England, actually in his book on diseases of the liver, he described fatty liver. He associated it with sedentary lifestyle and excessive fatty food. He was the first to say that on histology, we have to see fat in more than 5% of hepatocytes, and recommended abstinence from fatty food, sugar, alcohol, and recommended also increased exercise. And these are some of his drawings where he shows the black are the fat globules within the hepatocytes. In 1858, Rudolf Virchow, in his treatise on cellular pathology, Virchow is our main, most important father of pathology, because he started the concept of cellular pathology, that everything starts from diseases in the cell. So he also had a diagram of hepatic cells with accumulation of fat, as you can see in the upper photo. And he made the speculation that fatty liver would be cured by freeing hepatocyte from fat, whatever resmetirom is doing today. In 1861, we had the first microscopic descriptions properly of steatotic liver disease, and you can see these old photos, that these are from 1861, and they show really nicely the hepatocytes with tiny vesicles of fat, and this cut surface of a yellow parenchyma. And in 1861, the same physician wrote about the fatty infiltration and the fatty degeneration. He actually saw fatty degeneration close to inflammatory infiltrates and fibrosis, exactly as we define steatohepatitis today. So in he saw two different types of disease: fat infiltration, probably the current Mazdelt, and fatty degeneration, which is the steatohepatitis. And also these were described by Le Reboulet, who saw the first balloon hepatocytes, described them as cloudy swelling. These are the balloon hepatocytes, we will discuss about them, which make us recognize that the patient has mass and not muzzled. It seems that I cannot progress the slide. Maybe people, oh, oops. Sorry, here. Okay, thank you. And some clinical associations, these always come after the pathologist, of course. Charles Matchinson from England in 1867, he described painless hepatomegaly without jaundice. This is what happens to the patients even today due to excessive food intake and sedentary life. And from in the US, already they described healthy females, that it was more present in healthy females, precedes cirrhosis and treated with diet and exercise. And also in 1884, it was the first association of steatotic liver with diabetes mellitus. And association of steatotic liver with obesity was first described mainly in 1885 in the modern era. It took us 50 years to have a nice histological description of steatotic liver disease in diabetics. And also Charles Connor thought that it was related to cirrhosis. So even if we think that we are many years back, there was a kind of thinking about the steatotic liver pathogenesis, and what they thought between 1850 and 1917, they had realized the disorder of fat metabolism. There was increased delivery to fat to the liver. This increased intrahepatic fat, and also there was increased lipogenesis. You can see the names of the people who described this and the years. So it is an old disease, actually. We just started to realize it later on. Decreased fat oxidation mentioned since 1938, and decreased secretion from Virchow in 1858. So there is a timeline of major developments in muscle as we have it until 2000. I described with pictures the first years because I think they are important for us to realize how medicine evolutes and some things that we rediscover things. And we see them in a different eye, but we come back to where we started. The things mainly changed in the 1950s when we had the introduction of the liver biopsy. So we could stick a needle to the liver and not see just autopsy liver and see how the liver is in live patients. And Thaler, in his biopsy series, he used the term fatty liver hepatitis. And in 1980, this is the time where a pathologist, Ludwig, he used for the first time the term non-alcoholic steatohepatitis. In a famous paper, now has more than 4,000 citations, he saw this disease in patients, mainly female with obesity and diabetes, and that was a study on autopsy liver. So he actually rediscovered what we knew hundreds of years, at least 200 years, but he gave it a name and recognized it as a disease, proper disease entity. And in 1999, we have this seminal paper from Matteoni and collaborators. And this is when we recognize what made the difference, what helped pathologists to recognize what is muzzled and what is mass. And this was the balloon hepatocytes. So what Matteoni did, they followed up 100 patients. They had biopsy, and the initial biopsy baseline, we followed them up for 10 years, and they saw how many of them had cirrhosis. And they classified muzzled or NAFLD, as it was called at that time, as type 1 when there was only steatosis in the liver, type 2 was steatosis and inflammation, type 3 there was additional ballooning, and in type 4 there was in addition ballooning and fibrosis. And these two categories were the patients who progressed to cirrhosis in up to 20%, 25% of the cases. And this is how we started recognizing ballooning as an important feature for the diagnosis. Of course, you can see that there are some steatotic liver patients, that they progress to cirrhosis only having steatosis, but they are very, very rare. We recognize them today as well. And then in the same year, we had the first scoring system for NAS, and that was from Brandt in University of St. Louis University in Missouri. And this brings us to today. What are the current concepts on muzzled pathology? So we agree that in adult muzzled, we are not going to talk about children today because they may have different appearances under the microscope. The adult muzzle has a zone 3 predominance. So we see most of the fat surrounding the terminal hepatic zone. We have 3 zones in the liver. This is the portal tract. And zone 1 is around the portal tract. Zone 2 is the middle zone, and zone 3 is around the terminal hepatic vein. So we need to see the right pattern where the steatosis is. Because if we see it mainly around the portal tract, then this means that there may be a drug etiology, for example, or some metabolic diseases do so, or some steatotic liver disease in children do so. And for diagnosing steatohepatitis in adults, we need a constellation of findings. In addition to inflammation in the parenchyma and steatosis, we also see the hepatocyte injury in the form of ballooning. These ballooned hepatocytes are enlarged, have a size more than 1.5 to 2 times than the other hepatocytes, and they have this clear cytoplasm or flocculent, as it is said. It's not as white as the fat. You see there is some difference. It may have microvesicular steatosis sometimes in there. We recognize them from the round shape, the size, and what there is into the cytoplasm of the cells. Of course, there are some discrepancies on how exactly to recognize some intermediate ballooned hepatocytes of an intermediate size. And of course, another important concept is where fibrosis starts in mass. And fibrosis starts around the terminal hepatic vein when we see the steatotic liver starting and when we see the inflammation starting. And it is a special type of fibrosis that it is pericellular, surrounds the cells, and it is sinusoidal. It doesn't start in the portal tract, but we will see later that it may get there. So right now, liver biopsy is the best standard we have. It's not the optimal, but it is the best standard we have for diagnosing steatohepatitis, for grading the activity of the disease. That means grading steatosis, ballooning, and lobular inflammation. And there are 2 main systems that we use. The most popular is the NAS-CRN from the US, from the National Institutes of Health. CRN is the Center for Research for NAS. And this activity score semi-quantitates these 3 main features, steatosis, inflammation, and ballooning, and you get a total score from 0 to 8. And the SAF activity score, introduced about 7 years later, takes steatosis out of the picture because we realized that it was such a marker of activity. It's just an indirect marker of how the liver is susceptible to the rest of the injury and only scores ballooning and lobular inflammation. So these are 2 pictures of an early steatohepatitis and a more severe steatohepatitis, and this mild form, which is still muzzled because activity has only 1 for inflammation. There are not many ballooned hepatocytes. The score is NAS3, 2 for steatosis, 1 for the inflammation, and in SAF would be only 1. And this severe steatohepatitis gets 7 points because it gets 3 from steatosis, 2 from ballooning, and 2 from inflammation. And the SAF is an A4. We mainly use for the clinical trials the scores of the NAS-CRN from the 0 to 8, also semi-quantifying steatosis. Also, we need to remember that the natural history of muzzled is dynamic. So at any certain point, the patients may come back from steatohepatitis to steatosis with inflammation or simple steatosis by changing, for example, diet habits or exercising, and then get back to steatohepatitis. And on the basis of all these features, we have fibrosis. So fibrosis does not develop the same to all patients. There are some early fibrosis and some late fibrosis. Some go to fibrosis earlier and cirrhosis, and some other people not. And we don't know exactly why there is this difference. So the rapid progressors are about 20% that can get from early fibrosis to advanced fibrosis and cirrhosis. And there are also slow progressors. So there is a non-linear fibrotic phase. And the most important prognostic marker for all-cause and liver disease mortality in MASLD patients, independent of confounders, is fibrosis. So we are mostly also asking, in addition to the diagnosis of steatohepatitis, to say and comment on the stage of fibrosis. So with a liver biopsy, we can evaluate the extent of fibrosis and architectural remodeling. This is the staging. So F1, we discussed, is around the terminal hepatic vein. This is the first stage. Stage 2, we get additional periportal fibrosis. This is F2. F3 is when we have bridging fibrosis. There are bridges of fibrous tissue connecting terminal hepatic veins with portal tracts or portal tracts between them, or central, like terminal hepatic veins with other terminal hepatic veins. And most patients with F3, if they don't change lifestyle or if they continue their eating and other behaviors, or if they are diabetics, if they do not control the diabetes, they may progress to cirrhosis. Some people progress anyway. And in cirrhosis, we see the fibrous tissue, which is blue, surrounding whole nodules of hepatocyte parenchyma. So these are the 4 stages of fibrosis that we see in a liver biopsy. And moving on our timeline, what were the main developments until 2021? We have mainly learned more about the pathogenesis of the disease. We had the first genetic studies that show that some polymorphisms predisposed for steatosis and steatohepatitis. And we also had the main studies that started, that showed that fibrosis is important prognostic factors. And also in the last 4 years, 2017 to 21, we have large consortia, multi-center consortia, to tackle this disease and trying to find specific biomarkers, because the patients are too many that have muscle. We cannot do biopsies in all of them. We need some biomarker that can help us identify who has steatohepatitis or who is going to progress and who has fibrosis. So what we knew in 2010, we realized the central role of lipotoxicity in the pathogenesis of NASH. The scheme has not changed much from the 1870s, but we also know that there are adipokines that are secreted by the visceral adipose tissue that may modify and make the disease appear and progress. We realized that lipotoxicity, some of the bad fatty acids are doing to the liver, not all fatty acids are bad. And also how this pathogenesis underlies the features that we see in the liver biopsy. We also realized that the gut and the microbiota were important for the disease by releasing endotoxin, and oxidative stress and ER stress are important for the hepatocyte injury. And of course, genetic predisposition, this polymorphism, or the PNPLA3, was the first that was discovered. About 20% of the population may have the polymorphism that makes you be more susceptible to steatotic liver disease, but is also related, as further studies have shown, and we will see it with more steatohepatitis and also hepatocellular carcinoma. And we also learned about the multiple parallel hits, so everything is happening at the same time in the liver, that they're coming from the gut and also from the adipose tissue in the abdomen. So visceral abdominal fat is very important for the disease, and they promote liver inflammation, and there is a cascade of events that creates steatohepatitis, and then we have fibrosis. So in 2024, the extra thing that we know, we have realized more about the gut adipose tissue axis, microbes, their metabolites, and inflammatory mediators fuel the metabolic disease, the metabolic steatotic liver disease in the liver, and also adipose tissue in the abdominal cavity. And we have a lot more gene polymorphisms that usually affect lipid metabolism, and these have different types of effects on the disease. And we have also a protective polymorphism, the HSD17B13, which I will talk more next. When we try to discover drugs for MASLD, I mentioned we didn't have any approved until this year. Many clinical trials are taking place as we speak now, and all of them need liver biopsy because the endpoints to say if a patient has responded to the drug in both phase 2b and phase 3 trials is to have NAS resolution under the microscope without worsening of fibrosis. And in phase 3, you may have, or at least 1 point improvement, 1 point in this score that I mentioned on the NAS score, the NAS stage on fibrosis with no worsening of NAS. And similar endpoints are for cirrhotic NAS. So clearly, because most of these trials have not been successful, many patients could not get to the trial because the central pathologist, the initial pathologist, has said that the patient has steatohepatitis, and then when the patient went to be evaluated if they will go on to the trial, then the pathologist of the trial, who have seen a lot more cases and are more expert, they realized there was not enough ballooning, so there was no steatohepatitis. So in order to solve the problem with having more patients, we're taking drugs so we can follow them up, and also to make sure that how we select the patients for liver biopsy or how we select the patients for the trial, we need to find, and we already have some non-invasive diagnosis for MAS and fibrosis in MASLD. So there are combined scores that are based on clinical and laboratory data or serum biomarkers. You see the list here of the serum markers, the direct serum markers, and the combined scores. The NAFLD fibrosis score and the FIB4 score are the more that they are used. And also we have transient elastography and other imaging methods that can measure the elasticity of the liver, how much fibrosis has the liver. And also we have other methods like magnetic resonance MRE to assess liver fibrosis. The transient elastography is easily done and is in many centers now. The other methods like the MRE should be done in very specific centers, although it is the more accurate. And we have sequential combination of imaging with scores. So we try different things to find out of this cohorts of patients with MASLD who has steatohepatitis, who has advanced fibrosis mainly. These are mainly useful in primary care to select patients at low risk of advanced fibrosis, so we can exclude patients in primary care that do not have the advanced disease. We're more accurate in that with the biomarkers. And they also can predict long-term outcomes in MASLD, and they can reduce the need for liver biopsy because we don't do it in patients who have low risk for advanced fibrosis and they do not have other risk factors. They can reduce it, but cannot substitute the need of a liver biopsy until now. So the large consortia tried to identify and validate MAS biomarkers. One of these was the Litmus consortium. It didn't start with the Litmus. We have two other consortia since 2010, the FLIP and the EPOS, that led the discovery science in order to focus to the biomarkers. And Litmus is a big European project with 53 partners from 14 countries. And I had the privilege to to lead the pathology part. So we've seen more than 2,000 liver biopsies. We are a group of liver pathologists, 10 of us, looking at every biopsy in pairs to make sure that we have an accurate NASH diagnosis and muzzled classification. And the main role for the pathology was to be the backbone, and all the biomarkers would have correlate and would be compared with the liver biopsy as our best standard that we have. And this was also litmus and the other biomarkers were also applying the biomarkers to be those that were proven successful or better than the others for approval again in the FDA and the European Medicines Agency. So the Litmus is the European project, and NIMBLE is the project in the US. They are just large multi-center projects, and they are quite similar. As you can see, they all have diagnostic, prognostic, and monitoring of these patients. They try to evaluate better imaging and also blood-based, mainly blood-based biomarkers. The additional element that Litmus brings is the capacity for biomarker discovery from ANU. And also we had a lot of animal models that we tested, but I'm not going to talk about them. And we could do reverse translation to preclinical drug development pipelines. So both of them have a shared focus in the generation of the data that we will need to support the deployment of non-invasive marker in clinical drug development studies, and also at some point to routine practice, we hope. But the validation of by expert liver pathology interpretation or scoring was the important thing to do in order to have the best biomarker. So moving into our timeline, because I really like this journey through the centuries, but now the years passed very quickly and we discover things very quickly. So in 2023, we had the first biomarker consortia results that I will show to you. In 2024, we have the approval of the first drug for MASH. And what did the Litmus and NIMBLE consortia found out in these 5-year and even 6-year studies that they did? They had similar target conditions at risk MASH for Litmus, so more advanced MASH with NASH score more than 4 and fibrosis F2 and more. And for the NIMBLE, they only had patients that had NASH score more than 4. And there was also a significant difference in the acceptable performance threshold, 0.8 for Litmus, a higher threshold for the biomarker performance, and 0.7 in NIMBLE. And the population was a bit less skewed toward less fibrosis and progressive disease in the Litmus and more to fibrosis for the NIMBLE. And if we do the comparative analysis of the blood biomarkers for at-risk MASH, these are the findings from Litmus. These are all histological characterized muzzled patients, 933, that had complete data. You can see among all these biomarkers that we tested, even some more new ones that were developed during Litmus, none passed the threshold of 0.8 for at-risk MASH for the diagnosis of steatohepatitis. While for advanced fibrosis, luckily we had 2 biomarkers, the SOMA signal, which is a very complicated biomarker incorporating also some molecular data and some difficult to measure in routine markers, and as a more simple system, the ADAPT that has procollagen 3 and platelets and diabetes and AIDS. Diabetes, not AIDS. So these 2 had passed the threshold as more reliable for advanced fibrosis. And if you see the results from NIMBLE, because, and these are with the orange color, we see that quite a few biomarkers passed the 0.7 threshold. And when you will read the paper, you may see this thing that the results are different is because of the thresholds that we were using. And these 4 with the orange color proved the most reliable, the FIB4 fibrometer with elastography together, the ELF and the Procollagen 3, which are similar to what we found in litmus. So we're very good that we know that the results are similar across the 2 sides of the Atlantic. It's only a method of how we evaluated the results that differs, because this is the lower threshold. And if we had the 0.7, we would have in litmus also some more biomarkers accepted, but we were more strict, as it shows. So the journey that we do to find more drugs that are in phase 2 and 3 development is based on what we know about pathophysiology of MASLD, as I explained it. So for every step of those that we saw in the previous slides, we have different, we see all the names of the different drugs and where they target within the liver. And what happens today, and this is a very nice review from Mazen Noureddin in just a few months ago in Hepatology, where you can learn more about this. This is the resmetirom, was approved recently, and we have also the PPAR agonists that seem very promising, and also there are semaglutide, which is a different type of drug targeting GLP-1, and fibroblast growth factor receptors. These seem to be quite promising now, but of course there are ongoing studies in an earlier phase with other different agents. And in moving in our journey, what we have now and in the future. So we now started omics study, because omics studies to reveal new therapeutic targets. And we are moving towards to have artificial intelligence applications in MASLD. And I will show you some of the results of litmus, where we use liver biopsy in order to search for therapeutic targets and in this complex landscape of MAS pathogenesis. So how can we use liver biopsy to find targets with tissue markers? Well, at the beginning, we had we started with immunohistochemistry, and those balloon hepatocytes, we found that they secrete, we meaning the whole medical community, but it was mainly work done by researchers like Anna Maydill and others, who found that balloon hepatocytes secrete by themselves profibrogenic factors like sonic hedgehog. So we can see the sonic hedgehog by immunohistochemistry, and we can identify balloon hepatocytes. It could be an objective marker of balloon hepatocytes. Or also we know that from the studies from Caroline Lackner, that balloon hepatocytes lose keratin 8 and 18. So this is a positive marker, the sonic hedgehog, and the keratin 18 is a negative marker. Cells that lose the keratin are ballooned. And it's interesting to know that that's why we have the pericellular fibrosis around the balloon hepatocytes in zone 3, is because they secrete their own profibrogenic factors, and we see the fibrosis going all around the cell. We're able to visualize inflammation. We know, this is from the PhD study of GAD in Australia, that the first inflammatory hit in NAFLD, in MASLD, that we see under the biopsy is presence of macrophages in the portal tract. And then we have all the other inflammation coming. And there are B cells, T cells, subtypes of T cells, also shown by immunohistochemistry. But now we have molecular pathology and all these technological advances of high throughput RNA sequencing, single cell genomics technology that we will discuss, and some artificial intelligence pathology-based applications. And we consider this as part of molecular pathology. What we did already in EPOS in the previous project that we had, and we moved with this through the litmus, we did a transcriptomic profiling across MASLD, and we found gene signatures for steatohepatitis and fibrosis. We used a discovery cohort that was very well characterized on histology of simple NAFL and NAS. And then we did high-throughput RNA-seq on these liver biopsies, on 206 of them. And we had also a validation set, and we did some biomarker validation when we had the first results with some initial proteomics. So what did this transcriptomics showed? Showed that there were two, in the unsupervised analysis, so two clusters in these muzzled patients. We could identify through the gene signature who had simple NAFLD and who had more advanced steatohepatitis. Actually, you can see that these are the numbers of the genes that were found in this, were expressed in every set of patients, and 43 were the same that were both that could recognize between NAFL and NAS, and the same in another group of patients that had also already NAS and fibrosis. So this complemented, and we identified 25 genes associated with fibrosing mass. The most important of them were the AKR1B10, and also we can see the GDF15 and the HSD17B14, which is similar with the one that I mentioned before about the polymorphisms recently discovered, that is protective. It is a protective polymorphism. And using this signature, when we applied it to the validation set, we can, only using the signature, identify the patients with fibrosing, progressing steatohepatitis in human muzzled with a very good accuracy. And we could also see, based on the tissue, based on the findings that where these genes were expressed, which were expressed in epithelial cells or macrophages, but we couldn't recognize which one was where. So this could highlight where were the different intrahepatic cell populations. And as pathologists, we always go back to the tissue. And actually, we could see that this was happening already when we do simple immunohistochemistry with the KR1B10, for example, from NAFL to NAS F4. You can see how the KRB110 increases. And the same is more evident within other little cells for the GD5 and in macrophages with this other marker. So it helps us going back to the tissue and see where we start from and how we can then use less complicated technology to again study a tissue biomarker. And regarding the 17-BHD13, it was recently discovered that it is a hepatic retinol dehydrogenase. So it is involved in the metabolism of retinol together with AKR1B10, and it takes part in this biochemical pathway. And we can now localize it in lipid droplets within cells that they have it. We can see in this with a green color. And again, this is a stain that stains also the perilipin around the fat vesicles green. So these are a protective variant, is related with reduced inflammation, ballooning, and fibrosis in muzzle. And there are some targeted therapies in phase 2 clinical stage right now regarding this biomarker that we could identify on the tissue as well and in the cells. And we moved on proteomics to continue this work and see how the serum proteins may change and if we could do the same work using just sera of the patients. But again, we had to start from the liver biopsy, the RNA-seq, and take also plasma serum from all these patients, and do proteomics in more than 5,000 proteins and see if there are differences between the groups. And stratification was based on advanced fibrosis and high activity score. Using this stratification based on the liver biopsy, we could identify the proteins that about 30 of them that were differentially expressed between the NAS, those cases that had more activity and those that had more fibrosis. These are the top 50 more significant proteins associated with fibrosis and increased activity. And going back to more research, we could see which of those were more related to the presence, to be more protective or not related to the different colors that we may see here. These are very small letters, but I have highlighted for you that again, some of the already known culprits are here, the EKRN1B10 and also the ADAMTSL2, the TREM2 in the macrophages. So these are biomarkers that are differential expressed, could be targets, new therapeutic targets. And back to the tissue, the EKR1B10, this is NAFL, and this is NASF3. It's a lot more increased, and the same happens when we do this with more scientific ways, with all the columns and bar, it is increased in all phases. Talking about macrophages, which are one of the most important cells in the pathogenesis of NAFL, doing a transcriptomic analysis of 170 muscle biopsies, we tried to see the macrophage scavenger receptor and the differences that they may have in the different types of the disease. First, we could see that it was localized, of course, in the macrophages. And then these are the immunohistochemistry who shows the marker in the macrophages and how it changes between F1 and F4. It seems to play a critical role in lipid-induced inflammation and could be also a potential therapeutic target for muscles. And of course, every 2024 study should have some special proteotranscriptomics. And this helped us, though, to identify that all these macrophages are not the same in at-risk mass patients. So we can look under multiple markers in the same section. And we devised the areas where there is steatohepatitis, where is parenchyma with no steatosis and portal tract. And in these three different areas, we had different presence of macrophages and different factors that could be increased, like CCL1-19, more increased in portal tract, the MSR1 more in the areas of steatohepatitis. So I wonder if this could be also a strategy when you do a trial for MSR1, go to the biopsy and see where you have most of the injury and stratify, select those patients for the trial in order to have better results. And some cell phenotyping. These are all work that we did with Olivier Gauvaert from Newcastle. We presented last year at the EASL Congress, and the different colors, the yellow show the CD163 for macrophages, CD68, different type of macrophage, so the blues are in other areas, and the pankeratin is everything that it is white, the epithelial cell. So we could see the different types with this special technique. And I will finish with some data on AI in muscle biopsies. So now we are moving, in order to help us with a big workload of biopsies for the clinical trials, to have some help from AI. So already, it seems that in the near future, these trials will be supported by AI methods. So there are already some fully automated software applications, a very nice reviewed this one, that can quantify steatosis, inflammation, fibrosis, ballooning. This one is the one that is already published in hepatology, and it works with stained tissue. And I will not go into the details, but it was claimed in this paper that it was identified with good accuracy compared to the pathologists, the different features. And there are also advanced microscopy techniques like second harmonic generation and two-photon excitation fluorescent imaging, where we use an unstained section. We we can see we take the fluorescence of the unstained section and the collagen as well, which autofluoresces. And this could be transformed through a laser beam to a picture. And this is the picture you get under the SAG TPF microscope. Everything that is green is the collagen, red is the liver tissue, and it can identify tiny, delicate fibers of collagen that we cannot see with the histochemical stains. And also they can identify some ballooning, not with a very great accuracy, but this was the first study that we did where we taught the artificial intelligence system how to recognize the ballooning and then try it itself. But none of the studies that these are mentioned, they have not been validated. So there are these softwares that are being used, but they have not been validated in big cohorts in a large number of patients. But what we could see, and it's interesting in a recent paper, is when we quantify, for example, in this trial with tropifexor or TXR, this drug, when the pathologists saw the slides and compared between placebo and the cases that had taken the drug, we can see that we couldn't see much difference between the different doses of the drugs and placebo. So this trial would not show the clinical endpoints that the FDA wants. But if you do the more sophisticated technique of second harmonic generation on the unstained section, this could identify indeed difference, the blue colors, compared up to 83% difference in staging, in the F3 stage, and compared to the placebo. And this was because they could identify regression in the sinusoidal fibrosis, in the thin sinusoidal fibrosis. And this was important for the trials. It means that we need to get more data from mast fibrosis dynamics and have more insights on treatment-induced fibrosis regression. So also using the AI, we can help agree with each other for the fibrosis score according to other studies. So AI would help the pathologists reach a next level of recognition. But again, when we validate all this AI properly. And the last thing that I will show you in this long journey is that we now use we can use unstained tissue section to automatically stain. So we just need one section. We can do all the stains artificially, right? Virtual stains. You see that's the virtual stain, and that's the real stain. There is no difference. And using these virtual stains, a group of pathology, including me in the group, actually we couldn't recognize which was the real stain and the virtual stain on the digital slides. And we taught the machine how to recognize with annotations what were the features, and it could identify some of the main features of steatohepatitis and give NAS-CRN scores. So we have come from 1843, Pierre Louis, who said that it was the most frequent and significant disease of the liver, which we know, to Maslow 2024, the most common cause of chronic liver disease worldwide, a long journey. But now we need to focus on establishing preventive method for the disease. We need to find better non-invasive diagnostic and prognostic biomarkers and develop more effective treatments for individuals with at-risk MAS. And thank you very much for your attention. And I would like to thank also my fellows liver pathologists who continue to play a central role in steatohepatitis diagnosis and biomarker development. And they will continue to validate these non-invasive biomarkers and assessing drug efficacy in the future. And that's a free link that you can use. It's free on the European Society of Pathology platform, where you can have an atlas of images and see how we score steatohepatitis. It's about 100 images, and there are guidelines for diagnosis and scoring. So thank you very much for your attention. We have a lot of years, even the modern years, to get to in order to have this in modern diagnosis. The important thing here, I think, is the the whole translation. So we're doing all this complicated research and we identify the targets. And I think the focus will be to make this more available, translating back to the tissue. So I'm not sure if we are going to do in all the patients all these special techniques, but if we manage to get all this knowledge back to the simple level of the tissue, if we still have liver biopsies, we will still be able to, at some point, use it in routine with simple markers, as I saw some of them. For now, where we do not even do the NAS scoring in routine reporting. Only if we are in specialized academic centers working with hepatologists that are specialized in the field. So, and we don't know also how the generalizability of this scoring in routine. So I can do it, another person can do it. We don't, in routine practice, in trials, we have done a lot of studies to see agreement among us and differences. We don't know what is happening in the real world exactly. There was only one small study. So I think it's important to continue doing this research in order to identify the therapeutic targets. If we manage to stratify the patients according to this, we may have better drugs. So in we say usually that steatohepatitis in alcohol, people with alcohol use disorder and for example, diabetics or obese people, it is similar. It is similar, of course, it's the same steatohepatitis, but it's not the same. So there are some features on histology that we can see that in people who use extremely alcohol compared to those that they don't. And that we never see in MASLD. So these are like veno-occlusive lesions. So this fibrosis in zone 3 becomes so increased that obstructs the terminal hepatic vein. And when we see that, or when we see a lot of extensive sinusoidal fibrosis as well, and pericellular, we suspect of some alcohol use as well. Of course, if we see more inflammation, there are some factors, but with only more inflammation and only more Mallory-Denk bodies, no, we can't do much. But this cholestasis is not seen in MASLD usually. So there are some features that make us suspect, and we write it in the report.
Mega mitochondria is difficult to see, is right?
Well, no, when
it's also very alcohol specific, but it's difficult to.
But we see them in MASLD as well.
You see in MASLD as well.
Because in both diseases, mitochondrial abnormalities play a role in the pathogenesis. I didn't show the megamitochondria. Megapitochondria, they are so big that we see them under the light microscope. So that's why we call them mega. And they are like pink globules. So we can see them. We just need to differentiate them for red blood cells that sometimes get inside the cell with the sectioning. Starting from the liver biopsy, we can see, we can recognize in most cases when there is extreme portal inflammation and raise the suspicion of a concurrent autoimmune hepatitis. I don't think there are any proteomics study to recognize between the MASLD and MASLD AI. But there are studies on using artificial intelligence that they try to differentiate very, very, very early. But usually, like for example, for a pathology lecture, we have a whole lecture on how to make the differential diagnosis between MASLD and MAS and autoimmune hepatitis, because MASLD patients, Dr. Londoño and Dr. Bartaler know better, may also have autoantibodies. So the clinical question is, this patient has MASLD or has autoimmune hepatitis, and increased transaminases and everything. So this is the reason we get a liver biopsy. And when there is, sometimes it may be difficult, but when we see more portal inflammation compared to the lobular, and more interface hepatitis, because you can see interface hepatitis in MAS, but when it is more than usual, we use that criterion to help. We always like to take, to help our clinical colleagues. We just don't just describe, but say that what we think that could help. There's the injured hepatocytes, these balloon hepatocytes. They used to be called undead hepatocytes or zombie cells, or they are in senescence. That's the latest data show that these are senescent cells. And when and sometimes, I think that at some point they die because we see a lot of apoptotic bodies as well in muscle. We never put apoptosis, cell apoptosis too much in our scoring. That's something that we could do for the diagnosis. And the hepatocytes that have fat inside, they have reduced regeneration, studies have shown, but they still, the hepatocyte can lose the fat quite easily if we stop the trigger. So that was shown since antiquity.
So thank you very much, Dina, for joining us on the liver seminars. It was a great pleasure to have you here. So I will see you all in the next liver seminar. So thank you all us for joining.